Good morning, everyone. I am Dr. Melvin Jo, Assistant Professor in Agricultural Microbiology, Department of Soil Science and Agriculture Chemistry, SRM Institute of Science and Technology. So today we are going to see about the gram staining of bacteria. This is basically, we are observing very closely. You can see two type of cell types. The main difference is the gram-negative cell wall has a periplasmic space and it also has an outer membrane. So due to the presence of this outer membrane, the membrane lipids will not get easily washed away when we are going to do the staining. This is basically the principle which we are going to use in the gram staining technique. So how we are going to have, why we need this gram staining technique? This is, gram staining was to be devised by the Christian Gram. He was to be working with the lung bacillus, and when he was to be working with the lung bacillus, he wanted to differentiate them into two types. One is the gram negative bacteria and the second is the gram positive bacteria. And the principle is like this. First is we are going to add the main dye, that is a crystal violet. Then during the decolorization step, the crystal violet may get washed away, as in the case of the gram negative bacteria. However, as in the case of the gram positive bacteria, it will get retained because they did not have the outer membrane. So they will be forming the crystal violet complex, and ultimately the cells will either appear purple color or to be red color. We will be seeing the staining technique. The first step is the addition of the crystal violet for around a period of around 60 seconds. After that, we need to wash it very thoroughly. Then we need to add the moderant. Moderant is nothing but grams iodine. Around 60 seconds you need to add. This should be followed by washing. Then we have to add ethanol. And the final step is safranin. You can see the two types of cell types. One is to be purple color and the another one is the red color. The purple color is to be the gram positive bacteria and the red color is to be the gram negative bacteria. So today we will see how this gram staining is to be done. The first thing is we need to have a very clean grease-free slide, and you need to take a very actively growing 24 hours old culture. And the first important thing is you need to make a very thin smear. That is very important, because once you make a very thin smear, then only you will be able to visualize the bacterial cells very clearly. So you need to have a very uniform distribution of the smear. So in order to get a very uniform distribution of the smear, our main thing is we need to have a grease-free slide. So these are the things we are going to have: crystal violet, iodine, decolorization, that is 70% ethanol, and next is the safranin. So basically we will be we have prepared the smear, that is to be very thin, and we are going to observe it in the microscope. Conclusion: today we have seen the gram negative bacteria. Thank you.